So I'm back in body, if not in mind. Late last night. Yeah, it's only 10 hours time difference to Finland, so we'll see how well I manage for the next couple of days anyway. In theory, I wasn't there long enough to get jet lag, right? So hopefully the last two lectures went okay. Good, yes, happy. There will be questions on them on the exam. I brought everything you need to know about Lambda. So just read this by Wednesday. You'll do fine for that part of the test. So not that critical. I did pull up a couple of notes for my lectures from last year that if we get to at the end of today, we can take a look at. But first, of course, the reason everyone's here is because we have our clicker questions for the day. First one is wild type Phi-X174 requires blank different scaffolding proteins. 0, 1, 2, 10, or 180. Okay, what are they called? B and D. The reason it says wild type, that is because you can also make a mutant that doesn't need B, but that has to be a mutant rather than the wild type. So for today, have that. Our next question for today is the rep protein of BSL-RDHV, my second favorite virus, is most similar to rep proteins from plant Gemini viruses, rep proteins from animal circoviruses, the A protein from Phi-X174, T7 DNA-dependent RNA polymerase, T7 DNA-dependent DNA polymerase. And if you get this one wrong, I can blame George. Ten, time to start guessing. 5. Okay, so yes, it is more similar to the rep proteins from circoviruses. These are the animal circoviruses. Gemini viruses are plant viruses that replicate in a very similar fashion. There are other RDHVs that have rep proteins similar to the circoviruses, or sorry, the Gemini viruses. The rep protein from A, which is then from Phi-X174, that's a very similar function, but the sequence is really quite different. It's a whole different family. So they're all different kinds of single-stranded DNA viruses that replicate in different ways. And then the DNA-dependent RNA polymerases and the DNA-dependent DNA polymerases, are because rep's got this really horrible name. It really has, it's the initiation of replication. It has nothing to do with the actual enzymatic process which is involved there. So B is our correct answer. And then the last one, which is lambda, and I can blame the video for this one, is when inside the cell the lambda genome is linear with repeated ends, linear with unique ends, a concatemer, circular, or degraded. Not what's inside the virion.
When it's inside the genome, it's also inside the cell.
Mm-hmm. Yes, it is.
And when it's
Go with your flow. Go with the gut. 10. So the comment I hear at the front, which I really like, go with your gut feeling on Stedman's questions. Don't assume that they're ridiculous trick questions. And the answer to your trick question, yes, what's in the genome? It's part of a circular genome because the whole chyle. Yeah, but it doesn't say it's circular itself. It says the lambda genome is. So yes, it's circular. Lots of these other ones are what happens to other different virus genomes while they are there. So those of you who need to disappear and would rather to study yourselves, adios. And now, this is what I was doing in Finland. This is the opponent. I was the opponent on the PhD exam, so think yourselves lucky I wasn't examining you. No, I won't be wearing this on Wednesday. So that was about half of what I was doing while I was there. The other half of the time had to do with this. But do you guys recognize that beer? I did not bring it from Finland. I was at the party and people were getting this beer. I was like, huh? Am I like sleep deprived? What's the deal? But apparently, Sierra Nevada is very popular in Finland. Go figure. And I even, this is our picture with my phone, so it even has the GPS coordinates. It really was in Finland. I didn't make this up. So, yeah, that's what I was up to for the last couple of days. So, that being said, this is, again, for those of you who haven't had my classes before, I just go over basically what was in the lectures I think are particularly important, things that I think I did a bad job explaining the first time around. This is no guarantee. This is stuff which is going to be on the exam. So it's just stuff. And please stop me because I'm going to go pretty quickly through this stuff. So if something that you don't really follow, please grab me as we're going through this. So basic story here, what's a virion? I'm trying very hard through this lecture to concentrate on virion as being what's on the outside of the cell. And of course, what's on the outside of the cell is only part of what's happening in terms of virus replication. Protein capsids, again, this is the protein that's on the outside. In some cases, you have envelopes and then the envelope proteins which are there. Why do I emphasize this? Because really it's all about the virus life cycle going from virion in the outside, but the genome that gets integrated on the inside, yes, sometimes it's circular, sometimes it isn't. Sometimes it gets integrated in the genome, which then makes more virions on the inside. Those virions then get released. And it's really this whole process of virus replication, which is what viruses are. They're everywhere. One of my favorite pictures here, of course, all those little dots, way more little dots than anything else which is there. What about the human genome? We are 8% viral, clearly very similar to these retroviral-like elements that are present in the genome. One could argue, and of course I do, that it's up to about 40% of the genome that's clearly related to these viruses, and then of course we're only 1.5% really human. So we're much more viral than we are human as far as this is concerned. Yeah?
If you were to ask this though on a test, we're sticking with the 8 to 10% for sure.
Okay, so the question here down at the front was if someone were obnoxious enough to ask a question about this on an exam, say Wednesday. By the way, it's not done yet, so it will be by then. And I've tried really hard to proofread it. Then, yes, I would be asking you about the part that nobody has any questions about being clearly viral. The other ones, I would not. So I could imagine two kinds of questions, but one would be what's really obviously viral and no one argues about. That's the 8 to 10%. And what does Stedman think is viral? That would be more like the 40%. So I'd be clear about that in terms of the exam. Any more questions about sort of general viruses? Again, virus ecology would be a wonderful course we could offer if we had twice as many faculty in the department and I wasn't teaching all these other classes. So how many? This is not so much about the messages on the side here. It's really about the plaque assay. Again, those of you who are taking the recombinant DNA techniques lab or are working in my lab know far too much about plaque assays. But the basic message is here that you have very small amounts of virus, huge amounts of hosts. So that means your MOI is really low because it's just the number of infectious viruses per cells. It's a very low number of MOI. Then you have a large number of host cells. The way you get this small MOI is you do lots of dilutions. Then you mix them together, let them grow, and then each of these plaques represents one virus infection, but it's gone through multiple rounds of virus replication because you wouldn't see a plaque if it were just one virus infection. So it's replicated, it's infected the cells around that, which have then replicated and infected the cells around that, etc. And then you have the agar, which you've mixed in here to prevent all of those viruses and cells moving around. So each of those plaques represents one virus infection at this low MOI. You use multiple plates and you take the average of all of those to get you how many plaques per mil you had in this diluted, and then you can back calculate where all of these are. This would be the kind of calculation that I would expect you to be able to do without a calculator. You should not need a calculator for the exam. Why do you do plaque assays? Basically, it's to get you the numbers here on the y-axis of one of these one-step growth curves. The way your one-step growth curve works, again, bug the people who are working in my lab about this. You do an infection now at a high MOI in liquid culture. So you haven't done any kind of dilutions here. Very high MOI, between 5 and 10. Why? So everything's infected more or less simultaneously. We'll get to the Poisson distribution in just a second here. And this again starts with an MOI of 10, PFUs per cell, starts 10, the black line here. Then all you do is with your batch culture, just let that sit and replicate for one full round of replication. And then you get lots of virus production. That's what you can see up here in the black line. And then all of the interesting stuff that happens here before you get infectious virus particles, which again is what you're detecting through your plaque assays here. All of the interesting stuff, production of proteins, productions of viral nucleic acid, productions of capsid proteins, then finally assembly of the virus particles. You can look at that here. That was a stylized version. Here are some real versions here. Start out with a high MOI that goes to 1. And the Y, do you get a 1 PFU per cell during this latent period, eclipse period? Okay, so if you think about what are you doing here? You're collecting a sample over time. and then doing a plaque assay with that. And so if you have an infected cell, what's that going to do? Your infected cells, each of those infected cells is going to be making one plaque because that will be where the virus genome is. So that's during this eclipse phase, you're always going to have one. So then that will grow very rapidly. It grows, of course, much faster than you would have with binary fusion. Also tells you that viruses are growing differently than the cells that they're infecting. You can do an extracellular lysis as well as just waiting for what's present on the inside, and that gives you this difference, extracellular versus intracellular. Extracellular particles are going to be infectious either way if you're a naked or an enveloped virus. However, the intracellular particles are usually not going to be infectious if this is an enveloped virus because it needs to pick up the membranes, usually picking up the membranes as they're being released from the cell. Everyone's favorite equation in virology, the Poisson distribution. If I'm going to ask you a question, I'll give you this equation so you don't have to memorize it. But you might want to think about how you would know about how you've got infected versus uninfected cells. But the main point here is that your virus-host interaction is a random particle interaction with large numbers of different particles. And so what it means is you can have, if you have an MOI of 1, on average, you've got one virus particle per cell. But if you think about the Poisson distribution, it gives you about 30% of cells that have 0 virus particles that are associated with them, and 70% which have more than 1. And so that's the reason to use the Poisson distribution to think about this. It's just multiple particles, both of what you're using for infection and not, or it doesn't really matter. You think about interactions between two different particles, two different things that are happening there. As long as it's random, it's going to be described by a Poisson distribution. Any questions about Poisson distributions, one-step growth curves, plaque assays?
Yeah.
Yes?
Could you actually just do one with the circumstances in which you give us a problem like this?
So if you look at some of the old exams, usually what I'll ask is at, for instance, an MOI of 3, what's the percentage of uninfected cells? And so only the way that you would do that is say, okay, the fraction of uninfected cells is going to be P of 0. So anything's got 0 that have associated with it. So you just plug in K here. K 0, 0 factorial is 1. M to the K is going to be your MOI to the K. That's 0. It's going to be M, so you end up with just E to the minus M. So in this case, it will be E to the minus 3 because we said it was a MOI of 3. So that will be the proportion of uninfected cells. And then 1 minus that is going to be the proportion of infected cells. Okay. Make sense? Yes? No?
How about like b e to the minus 3 equals, point whatever?
Yeah, so if you look at some of the old exams, I'll have those numbers in there. So you won't have to do those. Yeah, remember, okay, what's e to the minus 4? I can't remember that. So, yeah. Good, happy, unhappy. I had a question about this. Someone sent me an email about what Baltimore class is. Do you have to remember what Baltimore class 5 is? No, you don't need to remember what Baltimore class 5 is. That there are 6 general classes of viruses? Yes. And how you get those different classes of viruses and what they are is important. And so it's what is being packaged in your virion. So single-stranded DNA, double-stranded DNA, double-stranded RNA, single-stranded RNA, negative strand, single-stranded RNA, positive strand, and then these way funky guys here, the retroviruses, which are packaged as a positive strand but have to go through this double-stranded DNA intermediate in order to make the messenger RNA. People ask about class 7 or about class 5A, etc. Those are actually, as far as I'm concerned, and in fact most people are concerned, they're just subsets of this retrovirus because they're all replicating through a reverse transcriptase mechanism. Was that your question?
That wasn't a question, but I was just wondering, what's the real difference between the plus
Okay, yeah, so plus and minus strand. Plus strand is coding protein, and then minus strand is not coding protein.
So it has to be translated and then.
So for the minus strand, you have to replicate that. You need to make the opposite strand before you can get any protein production. So what does that mean about what has to be in this light blue box? You have to have some way of replicating that. So it's RNA polymerase. In the case of some of these retroviruses, it's also the reverse transcriptase. So there are enzymes that have to come in with your genome, as opposed to one of these guys, which are the positive strand. Those guys can be translated directly. Other questions? This is arbitrary, and we'll see there are actually some ambisense genomes, which screw this whole mechanism up. But don't worry about that for this exam. Virus structure, really two main, call them flavors, whatever you want, description you want to talk about it, in terms of virions. And so again, there's a separation between virions and viruses. You either have helically symmetrical particles or icosahedrali symmetrical particles, sometimes a mixture of the two of them. And the real reason for that is smaller is better. The genetic code is really pretty inefficient, particularly if you only use one open reading frame. And so you want to have as simple a way of putting together your structure as possible. If anything, the helical arrangement is the most simple arrangement for putting things together, because each of your individual asymmetric subunits, that's supposed to be represented by comma here, interact with everybody around it in exactly the same way. Icosahedra are fine if you have a regular icosahedron where you just have 20 faces or 60 subunits. There it is actually just as simple as this, but usually that's too small for fitting your genome in. And so usually you have to go through quasiequivalence to get to a larger size. Quick reminder as far as icosahedra are concerned, I didn't bring my deflated soccer ball with me here, but you have 5-fold axes of symmetry. That's your pentagonal axis. 12 of those, or 3-fold axes of symmetry here, again rotating 120 degrees. You've got 20 of those. You also have 30 of these 2-fold axes of symmetry where you rotate 180 degrees, you get the same thing back. It's basically exactly what's on here. This is your T equals 1 icosahedron because there are no places on this icosahedrali symmetrical particle where you have anything but 5 of the subunits coming together. So this is 5 subunits coming together. That's 5 units coming together. Here is also 5 subunits coming together. You go around and count them. That, of course, as I mentioned before, is too small in most cases for most viral genomes. So you do quasiequivalence. You actually make the soccer ball. You put in a hexagon between the 12 pentagons which go around the outside of your icosahedron. Here's the classic example, and this is really what you have with the soccer ball. Here at the 5-fold axis of symmetry, you have 5 coming together. Here's your 6-fold axis of symmetry, and then this goes back to your 5-fold axis of symmetry. And just a way to describe that is the triangulation number, h squared plus hk plus k squared. And that's just going from 5-fold axis to 5-fold axis. Difference between h and k is when you change directions. So h is one axis, k is moving on the other axis. People happy with t numbers? Putting together icosahedra, giving you something. Yes?
I guess I was just confused if like the wire model is sort of what it looked like. We used it in a previous lecture when we first went over it.
Mm-hmm.
Are you just assuming that each one is a 5-fold?
Yeah, so on a wire model, unfortunately, I don't have one here, but it would have pentamers and hexamers. And so the pentamer, you assume there are 5 subunits there, and the hexamer, you assume there are 6 subunits there. And so you'd count from middle of pentamer through middles of hexamers to go there. And that would be hexagons, again, in just that wire model. Make more sense? Yeah, it's often, I find, easier to use those wire models than something like this where you've got all these extra pieces on it. It gets rather complicated and confusing. If you start looking at cryo images, they get even crazier. So if you look at old exams, those cases where I have had those icosahedral structures, they'll all be wire diagrams. So take a look at them. Yeah, because there I find a lot easier to deal with. The exam I was on in Finland was one that was t equals 28, which was also really interesting. One of the things I think I did a really horrible job of explaining the first time around was these helically symmetrical particles. This is the case for tobacco mosaic virus. But what I wanted to mention here is sort of that the P number is sort of the equivalent of the T number. It's a way of describing how you put your virion together. And our P, which is the pitch, again, it's just a way of describing how these virions are put together. And all that it is it's the number of subunits that you have per full turns, how long it takes to get around, times the distance that you go between each of these turns that you go around in the process. And so if I give you for instance, number of subunits per turn times the displacement, you could give me the pitch. And then if you know how many nucleotides each of these subunits binds to, then you could also tell me how big the actual virus genome was for looking at one of these helical structures. And that's in fact how you can get some really nice numbers of either how big the genome is or how many of these capsid proteins you have that associate with them. So icosahedrally symmetric, helical symmetric virions. This brings me to our virus classification. We talked about the ICTV. I didn't bring my ridiculous book with me. The person who was defending their PhD thesis in Finland came up with a whole brand new virus family for these t equals 28 particles, which is really kind of cool. Whether I agree with her or not is a different story. But the basic message there was that if you want to think about relationships between all kinds of different viruses, particularly between very different viruses relative to each other, you're much better off looking at the structures of the capsid proteins themselves, because those are much more likely to be conserved than the actual sequences that are present in any of these virus genomes. And so that's basically what this message is about. And yes, my third favorite virus now, I guess, STIV. This particular capsid protein structure here is very similar to bacterial capsid protein structures, eukaryotic capsid protein structures, and that leads us to the assumption, which is a pretty big assumption, that there was an icosahedrally symmetric virion here with that kind of capsid protein present probably billions of years ago. So, but there's no sequence similarity that we can detect there. And so I think that this is a really good way of classifying viruses, or I should say viruses based on their virion structures. Other people agree with me, including this group in Finland. But again, that's only one way of classifying viruses. We've also got the Baltimore method and then ICTV, particularly involved in sequence similarities. So questions on classification, etc. Moving on here. Now again, this is still very more general. What do viruses interact with? When they need to find a host, they interact with receptors. Basically, anything that's on the outside of the cell can serve as a receptor. And we found examples for pretty much all different kinds of things that are out there. The main ones we've talked about are the immunoglobulin domain containing receptors, because those are things that love to stick to things, and of course viruses need to stick to things as well. We've also talked about some of the sugar molecules on the outside. Sialic acid is a classic example of that. Influenza binds to it. It's the very basis for the hemagglutination assay that we talked about before, but didn't review again now. So pretty much anything that's on the outside of the cell, but those are the main ones that we've talked about so far. Okay, binding to the outside of the cell is all well and good, but of course you've got to get inside the cell in order to be able to release your genome. So going way back to that cycle. Think about virus life cycle. The genome has to get inside the cell. How does that happen? Usually via receptor-mediated endocytosis. So binding to the receptor forms now an endosome inside the cell. Then there's membrane fusion, which happens for most of the enveloped viruses, that happens between the virus membrane and this endosome membrane. Or in the case of the naked viruses, often then there will be a lysis of that internal membrane on the inside, and then the virus capsid is released inside the cytoplasm. Of course, the genome still needs to get out of that capsid. And we'll take a look at that, particularly for some of the more specific ones, a little bit later on. But most of the times it's going to be receptor-mediated endocytosis. Once you're inside the cell, and that's best shown probably right here for fusion to the cellular and viral membranes, release of the nucleocapsid here. All of these viruses that are shown on here are getting themselves to the nucleus. Why? That's where all the goodies are. And most of these are going to be DNA viruses. So for your animal viruses that are DNA viruses, they need to get to the nucleus to replicate with, as always, exceptions. This is biology, so there are always exceptions. But most of these are DNA viruses. There are a few RNA viruses that we'll talk about a little bit later on as well. But that's basically because that's where the goodies are. If you need to have cellular DNA polymerases, all those DNA polymerases are in the nucleus. If you need DNTPs, so deoxyribonucleotide triphosphates, those are all in the nucleus. So it makes a lot of sense to get there. So of course you have to get to the nucleus, and that could be microtubules, could be other ways that you're getting there as well. Very often this is microtubule mediated. This fusion of two membranes is clearly something that's really important for enveloped viruses. How does that happen? Mostly it happens because of conformational changes. And so that was a lot of the videos that we finally got to look at in class, where you have a big change in usually the virus receptor binding protein. And the big change there is due to either direct interaction with the receptor or something else that changes as the virion is entering the cell. Classic case here is pH. So change in pH leads to a conformational structure of your receptor binding protein, or in some cases a separate fusion protein that then basically pulls those two membranes together. And once those two membranes have been pulled together, then you release whatever on the inside of them. In the case of this one here, this is the influenza hemagglutinin, releases the virus nucleoprotein complexes which are inside that. Dengue virus releases the genome which is on the inside as well. HIV releases the nucleocapsid. All of these basically have extremely similar structures here in terms of getting those membranes to actually fuse. Yeah?
What's making, we're still outside the cell at this point. Is it acidic outside of eukaryotic cells? I know often it's outside of bacterial cells.
Okay, so the question basically here is what are we looking at here? And so what we have here, down at the bottom, so what's not shown here is the viral membrane. So the viral membrane is down here at the bottom. And then this is the outside of the virion. This is the outside of the virion as you're producing the actual virion. This is the HA0, which you actually don't have until you're actually on the outside of the cell. What you have on the outside of the cell as a virion is this one right in the middle, HA1 plus HA2. And that's because there's a proteolytic cleavage that takes place here, takes HA0 and turns it into these two proteins. HA1 binds to the sialic acid, which is out here. That's what happens when you interact with the cell. So this is all extracellular.
Right.
But there's no change in the structure until you get inside the endosome. And that's where the pH changes. And so it's inside the endosome that you have a change in pH. That's when you get the fusion that takes place. And if you back up a little bit, that makes a lot of sense because you don't want to be fusing necessarily in the outside of the cell. You don't want to be causing all these holes that are happening outside the cell. It's much better to cause a hole when you're already inside the cell. That way the cell's not going to lose all of its goodies out in the environment.
I guess I was imagining this bringing the viral envelope together with the plasma membrane.
Yeah, and so in this case, it's not fusing at the plasma membrane, it's fusing at the endosomal membrane.
It's getting endocytosed and then.
Endocytosed, and then there's a fusion to the endosomal membrane. And there are certainly enveloped viruses that are going to fuse at the plasma membrane. But here, it's fusing at the endosomal membrane. And again, it makes sense for two reasons. One, because you can easily change the pH there. Change the pH on the outside of the cell, it's going to be a real problem. And then the second reason is, making, making holes in the membrane. Fusing the plasma membrane is a lot more dangerous as far as what else will be happening as opposed to the inside. Pore formation is a big problem in terms of the outer membrane because, yes, the virus genome is going in, but then there could also be stuff going out as well. And so it seems that pore formation is usually only happening if you have a really thick kind of cell wall, very hard to get in, which is true for what kinds of things we talked about so far? A lot of the bacteriophages have to do this because getting through those two membranes on the outside is a real challenge. And they also don't have most of them don't have nice internal vesicles that you can then use as well. Okay, other questions on entry? So now we'll move over to some of the individual virus families that we've talked about. The RNA bacteriophage, and the main message here is we're really simple. And we have the major problem that all of these viruses with RNA genomes have to face. How do you deal with transcription versus translation? And how do you get different amounts of any given protein which is present inside your genome? There's just 4 genes here. All that you need is a coat protein, how to make your capsid. You need to be able to make more of your genome, and lysis. So in theory, you actually only really need these 2 genes here, way to replicate your genome and coat protein. And in some viruses, particularly hopefully the ones that George talked about, some of these circoviruses, that's all you got. Capsid protein, a protein which is involved in replication. Here, there's a extra lysis protein and a maturation protein. Of course, you need these at various different amounts. How do you regulate that? All has to do with the secondary structure of your genome. So here is the secondary structure in MS2, and particularly these overlapping start and stop regions, which are right here. The main thing, of course, that you need is the coat protein. How do you make the coat protein? That's the one that's got the best ribosome binding site, etc. So the very first thing that happens is you start making the coat protein. As you make the coat protein, then you allow the replicase protein to be made, and eventually, once you have enough coat protein, then you block making more of the replicase protein. Once the replicase protein has started, it blocks making coat so that you can replicate the genome, because if you've got the ribosome and the replicase trying to move through your genome simultaneously, they're going to run into each other. And so you have to be able to separate those from each other. The lysis protein is made from, it's called ribosome shunting. So the ribosome not completely falling off of your RNA and restarting. You need very small amounts of the lysis protein. You need it much later on. We'll look at some of the other ways that you modify the ribosomes later on throughout the course. The only other protein that you need here is the maturation protein. Maturation protein is only made from newly synthesized genomes. You only need it at the very end of your virus life cycle, and that's because you have alternative secondary structures which are present there. But it's all about those alternative secondary structures for these small RNA phage. Bacteriophage T7, then the first DNA virus family that we've talked about, the podoviruses. They all have very similar structures to these. Particularly important here are these early, middle, and late genes, or immediate, early, and early genes. These early genes are what are going to be coding for what in general? Immediate early ones? Yeah, dealing with the host. These are changing the host in some way. So we've got modifications to restriction endonucleases here. We've got shutting down of cellular RNA polymerase. Why do you get away with shutting down cellular RNA polymerase? Because we have our viral RNA polymerase, and that's what's coded right here. That viral RNA polymerase we'll get to in just a second. Your classic early genes are involved in making more of your genome. Classic late genes are involved in putting together the virion. So really nice example of looking at all of these different genes. Virus entry for all the podoviruses, and particularly well known for bacteriophage T7, is this interesting transcription-regulated entry where you have promoters which are released, and the transcription pulls the genome in. First through the cellular RNA polymerase, which is what's here in green, and then later on our friend the viral DNA-dependent RNA polymerase, which is incredibly useful for biotechnology applications because it's single subunit, it goes like crazy, and it has its own specific promoter binding sites. So this brings the whole genome in, but then also is going to transcribe all of the genes that you require for DNA replication and for making your virion, putting all of those proteins together. And then finally, as far as T7 is concerned, and this is true for almost all of the bacteriophage, these are just the ones that we have really nice cryo-EMs for, the DNA is packaged here in an almost crystalline form. It's really just crammed inside the virion in such a way that there's practically no space for anything else which is there. And as I mentioned in the molecular genetics course, kind of an interesting aside here is that if you take this and put it in an electron beam, it's actually not that dissimilar to B-form DNA. So that was a good way that Watson and Crick were able to show that what they found, their structure was actually probably similar to life because you could find it in viruses as well. 5X174 are best understood of the single-stranded DNA viruses, has this very strange genomic structure with all of these overlapping open reading frames, here particularly in the non-structural proteins. And what that tells you is that these things can undergo huge amounts of mutation because if you have overlapping open reading frames, changing one nucleotide could actually be changing 3 different proteins in 3 different ways. So when you're going to have these overlapping open reading frames, they're almost always going to be in non-structural proteins. And as we mentioned in the clicker question today, the 2 main proteins here that turn out to be non-structural because you don't have them in the final virion are B and D. These are internal and external scaffolding proteins. These are the ones that you actually need the most of. If you look at transcripts here, most transcripts for B, most transcripts for D, and then your structural proteins down here, F, G, and H, less transcription of these, less of the proteins, but also no overlapping open reading frames. So these, the structure is going to be much more important. You have to keep this conserved so you can't get away with overlapping open reading frames for these guys. The whole process of putting 5X174 together, also extremely well known, where you have these pentameric structures that get put together with the internal scaffolding protein. So let's see, these 5 membered structures that get put together, both of F and G. These now come together with the external scaffolding protein, giving you a procapsid. This procapsid has a different symmetry than your final capsid structure, probably allows the DNA to get into that structure. And then the really exciting thing here, which is not in the textbook, it's in these new articles, is what happens to the H protein when you have infection. When you have infection, this H protein comes together and makes this amazing pore structure and basically gives you a channel whereby you can get your single-stranded DNA into the cytoplasm of the cell. Here it's actually shown that the replication is helping pull your genome in. This is highly speculative. We don't know if that's really the case or not here. But it is clearly the case that single-stranded DNA is worthless in the absence of being replicated. So you have to have replication here before this single-stranded DNA can be used for anything. How does that replication happen? The first round is all about cellular polymerases. So the cellular DNA polymerase will bind to the single-stranded DNA, but doesn't bind to single-stranded DNA. It actually has to bind to double-stranded DNA. Where's that double-stranded DNA? It's a small secondary structure which is present in this stem loop right here. So this is what gets released on the inside of the cytoplasm. Cellular polymerase will bind to this. In fact, the cellular primase will make an RNA primer, and then you make double-stranded DNA. First, again, this is going to be one genome which gets released inside the cell. You have one copy of that double-stranded DNA. That then gets made into single-stranded DNA, and now we have the activity of the A protein, again, similar to what happens in the rep protein in the RDHVs that hopefully George talked to you about. What does it do? It binds to this double-stranded DNA, makes a nick in one of the strands, and that single-stranded nick serves as the primer for cellular polymerase. But now the A protein, supposed to be shown by this blob here, is bound to the 5' end where it's made that nick. And a nick, well, this is a reminder, that's just cutting the phosphodiester backbone. And it just makes a cut. It's hydrolysis of the phosphodiester backbone. So you end up with a 3' OH. Again, this is going to serve as your primer. You have a 5' phosphate. That 5' phosphate gets attached to a tyrosine on the A protein. Also true, which is happening on the rep protein. And that's why it's shown as a blob here attached to the end. The rep protein, in many cases, certainly for RDHV and the case in 5X174, also has helicase activity, so it pulls these two strands apart. So you can have the cellular DNA polymerase, shown here as the box, replicate its way around the genome until you get to the end of the genome, where this rep protein now takes the 5' phosphate that's attached to its tyrosine and hooks it onto the 3' end of the piece which has just been made. That gives you single-stranded DNA. That, early on in infection in 5X174, gets made into more double strands. Later on, this is what's going to get packaged. How does that packaging take place? Basically, is what we talked about before. You've got pentamers that form between F and G, helped out by this B scaffold. Pentamers come together, helped out with the D protein. You have the procapsid that then goes forward and becomes a capsid through final maturation. Questions on, 5X174, et cetera?
We're comparing the A protein to the RDHV's rep protein
Yeah.
a lot, but that wasn't the answer to what it's most.
Right. So the activity of the A protein is most similar to that. The sequence is more similar to the animal circoviruses. So, but the function is very similar. And so this brings up a more of a philosophical question, sort of, why? And are they actually really related to each other, but just related at such a level we can't see any kind of sequence similarity? Or is it some kind of convergence? You have single-stranded DNA, and somehow you've got to replicate it. But this is the way to do that. So those are just sort of two ways of looking at it. So one of the things that hopefully George emphasized, and if he didn't, I'm going to emphasize it again, is this difference between permissive and non-permissive cells, and the difference between permissive and susceptible cells. And so this all has to do with host cells. And the basic message here is there are two kinds of cells, susceptible and permissive cells. So susceptible cells are just something that expresses the receptor for the virus on the outside. That's all that it has to be a susceptible cell. A permissive cell has all of the replication machinery, and by replication machinery I mean whatever it needs to make the RNA or the proteins or whatever it is, not necessarily DNA polymerases here, on the inside of a cell. And so that particular cell, if you get the genome in, can replicate that genome and make virions. So to have a normal host, it has to have both. It has to have the receptor on the outside, and it's got to have all the machinery on the inside in order to be able to make those virions. And so a host has to be both susceptible and permissive. Now, this turns out to be really useful just in working with viruses, is you can have lots of permissive cells, and just by putting the receptor protein onto, or whatever the receptor is, usually using recombinant DNA techniques, you can make a otherwise non-host cell a host cell. And this is really useful for studying virus-host interactions, looking at various different receptors, figuring out what receptor actually is going to be involved with that particular virus. So if you've got a cell which works just fine on the inside, once you get the genome in, you can figure this out by just putting the genome in, again, through recombinant DNA techniques, electroporation, some other way, transfection, getting that genome inside the host cell, it'll still replicate. And so it's just an incredibly useful tool. And people will talk a lot about whether your cells are susceptible and or permissive for virus function. And this is just the example here for parvoviruses down at the bottom. Parvoviruses are a really interesting sort of subset, I like to think of the single-stranded DNA viruses. These are now viruses that have linear genomes as opposed to the circular genomes. But it's all about how you can replicate these genomes. This is what comes into the cell right here. DNA polymerases have to have primers, they have to have templates. So how do you have a primer and a template for replicating these single-stranded DNA viruses? It's a secondary structure which forms at the end of your genomes. And so it's this hairpin structure. Hairpin structure is great because now you've got a 3' end, you've got a template, your DNA polymerase can replicate its way through here, but then you have a problem. How are you going to replicate that other strand? And the way to do that is that these have complementary structures which can fold back up on themselves if you have DNA helicase activity, which is going to pull those two apart, and you have some kind of nicking activity, which, as it turns out, what kinds of activities does the A protein have, the rep protein in RDHV? Helicase, nickase activity. So it turns out that these proteins also have very similar activities as well. It's just here, these guys stick at this single-stranded DNA as opposed to being a circle, but they're very constrained by having these secondary structures which have to form at both ends in order to be packaged. And that basically here, it's the rep protein. I'm not quite sure why George put in here what rep does and what rep doesn't do, but I'm guessing he means it doesn't have DNA polymerase activity, but still has helicase activity, and I don't see the DNA binding activity, but also this endonuclease activity. Also the transcriptional activator, but that's true specifically for these parvoviruses. RDHV, again, our second favorite virus genome now because we don't actually have the virus or the virion. Circovirus, single-stranded DNA like replication initiation protein, again with an activity very similar to the A protein, or those non-structural proteins that you see in the parvoviruses. The big difference here is the capsid protein here looks like a single-stranded RNA virus. How the heck this happened, we've got some ideas, but we really don't know. That stem loop structure, which we can't see here, which we could see just fine on my computer before, is basically identical to the stem loop structure that you see in circoviruses. And so this is another reason why our RDHV looks more like, or so the rep protein is more like the circovirus's reps, because it's also recognizing the same substrate, this double-stranded RNA there. And so this is exactly where the mysterious messenger RNA joins the cell. Again, we can't see it here. The messenger RNA could be serving as the primer for second strand synthesis. Single strand comes in, you've got to make double strands. Or it could be for your single strand synthesis. And this is actually great because we know about as much about how this process works as what's really on here, as opposed to adding all of the other stuff. So, wow, okay, it could be anywhere. So there are questions about those single-stranded DNA viruses. Again, I wasn't here when George was here. I'll be asking him for a couple of questions, and I'll be making up a couple of questions. So there'll be some of both in terms of what's going to be on the exam. Yes, there definitely will be some. Okay, so let's finish up talking about Lambda. Again, it's all in the book. Here's the ooh la la centerfold from the book with all of the information that anyone would even care to know about this. But we'll go through this. This is now, again, not what, I don't have the video here, so I was going to pull up the individual pieces, but this is basically sort of the message which is there. Lambda, just like all these other viruses, has to get its genome in. What happens to the genome? It automatically becomes circularized once it gets inside. Then it can, in some cases, be integrated into the genome, and that's where it was when they found it originally. Or it can undergo this lytic growth where you have no integration and you immediately make virions. And then there's this interesting process of induction where you go from an integrated form to actually making these lytic forms. Again, how does lysogeny work? It's vertically inherited. How does that decision get made between whether you're going to be replicating lytically or undergoing this lysogenic cycle? The easy way to think about this is what happens with lytic growth. You get expression from these two major promoters, the left and the right one, going away from each other. And whether these lead to lytic growth has to do with termination. And so in the absence of the N protein, these guys terminate really quickly. Terminate quickly, you're going to end up having lysogenic growth. You're going to be shutting down virus replication. On the other hand, once you make N, and that happens through this PL promoter, N serves as an anti-terminator that allows transcription of all of these other genes, and most importantly here are the O and P genes. These are the classic early genes that are important for replication. So DNA replication that happens there. Then you make the Q protein. Q protein is another anti-terminator. In the presence of Q, now you start to make all the late genes. All these late genes are, not surprisingly, all the structural proteins that you need to make the virion. These structural proteins are mostly all over here at this end of the genome, which seems a little bizarre because here's our PR promoter that's leading out over here, but it's a circular genome. So here, these guys are directly connected to these guys. Again, circularized inside the cell. So that's what happens with lytic growth. Lysogenic growth is what happens when your host is unhappy. You make this C1 protein, which is the repressor protein, and it shuts down expression from both PL and PR. How does that happen? Has to do with binding sites which are present in these operators. So high amounts of C1, everything at this operator shuts down the PR promoter, but also the PL promoter, and shuts off this promoter, which is for keeping your repressor being made. On the other hand, when you're going through lysis, you're expressing this CRO protein. Hopefully you got this from the video. This is the control of repressor and other proteins, but mostly it's about the control of the repressor. In the presence of CRO, you shut down expression of the promoters which are making C1. In the absence of CRO now, you make a lot of C1. So it's all about the relative amounts of C1 versus CRO, which leads to either lytic expression or lysogenic conditions. What happens in lysogenic conditions? Yes, you shut down expression of all of these lytic genes, but you also turn on the integrase genes. These integrase genes will take your viral genome, circular now, integrate it into the host genome, which is also circular, so it's still circular, and I'll make that argument before. So now it's in the host genome with these two attachment sites. There's an attachment site in the viral genome. There's an attachment site in the host genome. Integrase binds to the two of those, brings them together, recombines them with each other. Then this gets reversed in the induction case. You make a specific protein called excisionase, XIS, which then causes this all to be reversed. So you end up then with a circular genome of your virus, which has been recircularized at the integrase site. Just a quick reminder, when it comes inside the cell, there's this cohesive end site, the COS site, that brings the ends of the genome together originally. Once you have made your genome, made all of the appropriate late proteins, now you have also made a concatameric genome, so multiple copies of the genome. How does that get made? It's through rolling circle replication as well, but not rolling circle like you get with your single-stranded DNA viruses. This is making double strands, but double strands with the cellular polymerase is going to make many, many copies. So your concatamer, and there you have concatamers also for T7. These are just genomes attached end to end to end to end to end, each of which has a COS site in it. That COS site gets recognized by the terminase protein. That terminase protein binds to the COS site, brings this genome to this head structure. And again, just like you have with 5X174, it's a prohead structure. It's not quite finished. Binds here. Then the genome gets pumped inside. And this is through a portal protein. Again, not unlike what you have with bacteriophage T7. Once you reach the next COS site, terminase cuts this off. You have one genome size, which is in here. This gets attached to a tail. The tail is made to exactly the right length because of this tape measure protein. Really amazing protein, just stretches out. It's got the exact length of the tail. The virus tail assembles around it, and then that is actually lost when you make it. So it's a scaffolding protein for a tail, as well as having scaffolding proteins for this particular head structure. So COS sites, are COS sites blunt? No, the COS sites actually have cohesive ends. And so they're overlapping sequences. So these are single-stranded end sequences which are made by the terminase. And we won't get into it exactly how long each of those are, but they're longer than restriction nucleases. Okay. Good luck on Wednesday. More questions? Email me. Yeah. What kind of scantrons? Big blue ones. Singles.